So I next would want to introduce Nick Butowski. He is the director of the Clinical and Translational Research in the Brain Tumor Center. He is really critical in being the liaison for not only the PIs of our Brain Tumor Center to bring what we're doing in the labs to the clinic, but also works very closely with the pharmaceutical companies to do that as well, and to really activate some very novel and exciting treatments for our patients. So Nick, you want to go ahead?
All right. Obviously, I appreciate the opportunity to speak today. I'm grateful to have so many of you here, and additional congratulations to Nancy and Bjorn for a fabulous agenda and meeting. This is my outline. I'm going to talk on translational research within the field, but in general as well, defining it along the way, discussing the translational pathway, where it fits into sort of the neuro-oncology GPS as we find ourselves, and hopefully have enough time to give you some examples from UCSF. Here are my disclosures. So during the 20th century, medical research has seen significant advances in basic science discovery. However, we haven't really witnessed a corresponding success in the common application of these advances into medical practice. Bridging the gap between such discoveries, i.e. what we know and have achieved at the bench, and the interventions, what we practice at the bedside, remains a challenge, really for many general and specific reasons that you'll learn about throughout today. To address this gap, the NIH made translational research a priority, forming centers of excellence in translational research as far back as 2006 as part of their CTSA program. Currently, there are 50 such centers throughout the United States, including, of course, at UCSF. The intent is effective translation of knowledge generated by advances in basic science research into new approaches for the prevention, diagnosis, and ultimately the treatment of disease, of course, including in neuro-oncology. But what really is translational research, right? So this slide gives you the every and entire answer and demonstrates that translational research, in translational research, scientific discoveries are moved along a virtual path from lab to real world practice, leading to hopefully improved outcomes, right? And it's basically divided into 4 phases. The first is T1, that tests findings from basics research into basically clinical effect or applicability, and involves what we would call preclinical or animal studies to phase 1 studies in humans. T2 research tests these new interventions in controlled environments to form the basis for clinical application, and in essence is phase 2 and phase 3 trials. T3 research explores ways of applying recommendations or guidelines in general practice and includes phase 4 trials, if you make it that far, and clinical outcomes research. T4 research studies factors and interventions that influence the health of populations, and in essence is social determinants of health research. And yet, how do we more narrowly define translational research for the purposes of a 15-minute talk? I think the simplest definition is that it's a process, again, of applying knowledge from basic science and clinical trials to techniques and tools that address unmet needs within a specific disease entity. It may be also helpful to apply it or contrast it with applied sciences, where translational research is undoubtedly team-based, right, and integrated amongst different types of experts, again, all focused on translating useful information from lab to doctor's office. So in short, it's bench to bedside team approach, but often is bidirectional with bedside informing back to bench. And none of it is simple or linear, and always begins with some sort of unmet need, and that is tackled in a dynamic team-based setting that's nimble and responsive and looks at a number of different possibilities, be it diagnostic devices, chemical agents, algorithms, and even business enterprises to address health conditions. So here's the translation continuum from discovery to early translation to late, and then dissemination and adoption. Obviously, drug discovery is the process by which a new candidate is discovered. Development is the process of bringing that new drug to market once the lead compound is discovered, and then preclinical research is done, leading to clinical trials, ultimately, hopefully, to regulatory approval and a new drug application. However, it's important to note that this timeline, really, this slide says 10 to 17 years, but actually it's getting longer. It's about 10 to 20. It requires 10,000 molecules to get 10 into trials and 1 possible approved drug. So all comers in medicine, that's about a 10% probability of success once a drug reaches a trial. That dramatically drops to 2% in oncology in general, and less than 1% in neuro-oncology for a variety of reasons that, again, you'll learn about in other talks today. 85% of this cost then is from failure, though it's not strictly failure if that information informs back from bedside to bench in some meaningful way. So long before a new drug can be imagined, scientists are certainly working to gain that basic mechanistic understanding of specific pathways that are subverted in a cancer cell, for instance. This is the discovery cycle. And the reason it's important is that and I'm mentioning it in one slide today, is that truly, I think very few of us have an appreciation or good understanding of it. It is what I say is a medical reconnaissance mission where scientists are seeking out to identify targets that might be attacked with a new drug, but also have to understand the mechanism surrounding it. And really, despite the advances in mechanistic understanding, we still don't know how they play an integrated role in many other pathways, and certainly how they can be specifically targeted in brain cancer in particular. And so testing of the expanding number of available biologic agents against millions of chemical entities requires highly sophisticated screening methods and is contributing to what I call computational optimism, which could also be called information overload, and certainly results in some measure of replication crisis. To get past this, in silico modeling is often done and gets us to lead compounds and ultimately to lead optimization, which produces a drug candidate that has promising biologic and chemical properties for the treatment of the disease in question. It's also important to consider that modern day drug discovery remains very capital and effort intensive, and that discovery is still a very lengthy, expensive, inefficient process with, of course, a low rate of new therapeutic discovery. For example, in 2019, if you can remember that far back, the research and development cost of any new drug was about $2 billion. In essence, basic discovery is funded by governments and philanthropic organizations, but often taken over at later stage development by pharmaceutical companies and more often by venture capitalists these days. To be allowed to come to market, drugs must undergo several phases of clinical trials, and of course, as I mentioned, most fail. Small companies play a critical role in this development timeline and cost, often selling their rights to larger companies that have the resources to run trials, but often this affects the timeline of development and ultimately the cost as well. So if you're looking to develop an agent, you also need to know about the disease you think that may benefit from it. This is, of course, increasingly complex with lots of therapeutic choices in oncology in general, except perhaps in neuro-oncology, but that also that means that we have ample opportunity to hopefully make progress. But to learn about those, you're going to need to consult experts in the field and, again, accept that the timeline is slow, iterative, and requires contingency management application. So narrowing in on the next steps after discovery, we're preclinical research in the dish or animals and toxicity filing for the FDA IND, clinical trials and regulatory approval. I'll touch on each in a bit more specifically. So taking a closer look at preclinical testing, you take a new chemical and molecular entity and get a pharmacological profile from it that will be beneficial to the drug's journey through its development. And is used ultimately to file for an IND. Here is where industry has particular strengths, and most development areas at this stage are based in biotech or pharmaceutical companies, though a fair amount can be done at academic centers, as we do in our brain tumor preclinical core through David Raleigh. They can also use specifications assigned in this stage to evaluate the chemical quality and purity of the drug, its stability, reproducibility, and so on, all of which results in the manufacturing process, again, which is very important to understand because it too really does affect the timeline of development and, again, the cost. The toxicology is, of course, done as part of the IND and usually arranged in discussion with the FDA as to exactly what models and how they are best suited for the agent in question. Links at the bottom of the slide will provide more info, and I'll not read it to you in the interest of time. At this stage and before testing in humans begin, an IND application is filed with the FDA, and when it's approved, trials can begin. From experience, professional mentorship here is absolutely critical, and that is you need someone who's done it before, including regulatory assistance and counsel on IP and conflict of interest. Such guidance can also come in part from the FDA in the guise of pre-IND application meetings, which are very useful. And I want to pause again to note that the main challenge to translational research is sort of something I've been getting at, and that is it's expensive, but that clinical trials in particular are expensive, have long timelines, and of course a complex regulatory component to them that is perhaps not always well understood. However, of course, clinical trials are the most important step in translational medicine, and of course they play a critical role in translating discoveries into new and better treatments. The efficiency of this process, of course, remains complex. It's required great team integration, and to incorporate modern methods of computational biology, genomics, proteomics, pharmacogenomics, and so on. You need experts within each of those fields, let alone to present personalized or precision medicine, as we just heard about from Dr. Clark. So unfortunately, and often in neuro-onc's case, industry's interest or even academics, they've not been necessarily comprehensively supported by the best tools, that is, the best models, which provide a good scientific backbone. And therefore, making judgments about clinical trial design in terms of endpoints and biomarkers becomes very difficult. And in fact, the unfortunate part here is if you look at success, a key indicator, if not the indicator of the translational process of success is biomarkers that are needed for translational prediction. In fact, studies indicate that biomarker creation or identification predicts safety and success from animal to man 80 to 90% of the time. And thus development simply needs to have a concurrent emphasis on biomarker development, be it in blood, tissue, or imaging, or the like. Ultimately, you write a protocol that includes a design team, and the phases are listed here. Again, in the interest of time, I won't list them or read them to you rather. And there are a number of ways to get a protocol sort of more quickly developed. A lot of these pathways you can read more about from the link at the top, but there are good ways to also raise continued venture capital. And if you're lucky and your trials look positive, you move on to a new drug application, which tells the story of your drug, including, of course, safety, efficacy, and directions for use. And then ultimately, once the FDA receives that, the team decides to whether it's complete and whether it gets approved. And the senior official makes a decision and gets back to you with any ramifications that may need to be looked at. And if you're approved, you work on labeling in the FDA and resolve any outstanding issues or questions. Haven't had too much experience, of course, with this in neuro-oncology yet, but I'm certainly looking forward to doing more of that type of approval work in the next decade. And so I thought it would be nice to give you some examples of trials that sort of highlight some of the issues I touched on in the remaining 4 minutes or so. The first example is a phase 1 convection-enhanced delivery of liposomal irinotecan using real-time imaging with GAD and recurrent high-grade glioma, which was both sponsored by NIH, R21, and Ipsen Pharmaceuticals. This was a first-in-human study and really came together from a translational and collaborative effort led at UCSF and built upon extensive preclinical development from our core and from support of an industry partner as well as the NIH, where sort of Neuro-Onc served as the leader or facilitator between all these parts to make them come together. The rationale for the agent is seen here. In short, due to the large size of the particle itself, when administered intratumorally via convection-enhanced delivery, it uniformly distributes throughout the brain and correlates linearly with the volume of drug administered. Furthermore, the drug itself was designed to release CPT11 slowly over an extended period of time with a half-life of 56 hours, which of course dramatically increases the temporal exposure of the tumor to chemo. And the delivery here was really optimized in rat, dog, and primate models with studies in the UCSF BTC core, comparing these routes and showing that CED was much superior. Another important translational advance here was the imaging part of it, the real-time imaging with the convection-enhanced delivery, meaning that the drug was also given with GAD and co-infused in the imaging suite. And this basically allows you to correct for the problems that are listed on the slide that we saw with prior CED studies. In essence, knowing where your drug went, correcting for any problems, and then correlating percent coverage of the tumor with outcome. There's not enough time to take you through how complicated things got with the intricacies of this study. I'll just simply note that we went from very low fixed volumes to variable volumes, optimized to cover tumor volume as best we could in a running conversation over years with the FDA using 1 to 4 cannula. And results-wise, we still have to look at a lot of data, to be honest with you, and I don't want to lead you astray, but in essence, variable infusate volumes were shown, the variable volumes where you had better tumor coverage have shown some promising results, and there's been some long-term survivors within this realm, of course, small numbers. And although the principle of CED here isn't novel, this study really does represent device and procedural translation advancement for the application of CED that increase precision, reliability, safety, and ultimately tumor coverage. And importantly, in the interim, several other clinical trials refined from this work, including Medsena and Istari, will have registrational trials in the year to come using similar tech. However, I think the variables of catheter placement, backflow, concurrent imaging, infusate volume, target volume, tumor coverage, AEs, and simply who leads this effort are all to be determined. And ultimately, I think the lesson here, translationally speaking, is it took about 15 years to do, with industry and NIH support constantly in an iterative environment, and that you can get it done. And now we look forward to the next steps, which include multiple infusions over time. I'll give you a very brief tour of one other trial that's up and running here at UCSF using enzastaurin for newly diagnosed glioblastoma in a phase 3 study versus standard of care with placebo. Enzastaurin is a protein kinase C beta inhibitor. Preclinical studies here and elsewhere showed that it was effective in terms of stopping angiogenesis and growth. And in fact, was used in a prior study here in a phase 2 trial, but without any enrichment strategy. And so when the drug changed hands from Eli Lilly to de novo, they looked back at their prior studies and found that DGM1 is a germline biomarker that might correspond to benefit from the drug, both in lymphoma, but also in our prior trial where we saw some interesting long-term survivors. And so this biomarker with a new trial will move forward in the study design you see here, basically very simple randomization, 1-to-1. You get radiation Temodar and enzastaurin at a better dose versus radiation Temodar and placebo. These are just the run-ins, nothing to really explain for these slides. And then the outcome, of course, is survival. And this is really recently just started in the past month here. And here's where I'll take a moment to let you read some of the translational concepts that I think will help neuro-oncology move forward. And remind you that the way of really looking at this is to think about the expression of doing the right thing. It's really hard to know what doing the right thing is. And in fact, I don't think we can know for sure. And so maybe we should do, or rather we should say, do the next thing. And if we do that, whatever it is, to the best of our ability together in a translational model, chances it'll turn out to be the right thing and contribute to the field. And this, of course, means as teams rigorously assessing PD, PK, parameters of drugs, optimizing subtype selection, which means biomarkers, and prioritizing the best drugs and combinations in the best settings of trials. So I hope the above examples demonstrated the operational process that led to considerable evolution and adaptation over time, i.e. translational research. Yet they also do show the key problems that led to the emergence of translational research itself and how they in large part still exist. That is the long time from discovery to use, and that more investment in biomarkers and enrichment strategies needed from earlier in development. Part 2 of this talk would certainly cover conceptual models and operationalized methods of translational research, but I'd be happy to give that talk at another time. And so once again, I thank you for allowing me to talk and for your attention.